A little bookkeeping and housekeeping first, everybody. And here is the first item. If you look on your blackboard, and ladies, as you look on your blackboard, this is basically what I just sent you PowerPoint, some of the PowerPoints from the same PowerPoint people like this. I'm unable to, larger ones with a lot of animations go over the size constraints that are associated with this Blackboard Ultra. We didn't used to have the problem, surprise, but apparently, so I basically had to email it to you and use it as an attachment. So you can follow along with me when I use them, but you can download them if you want to review the animations that are in them. The Welsh Hole ones fit, so you can see, and if you did not know, when we switched to Ultra, a lot of it had to do with switching to compatibility modes and ADA compliance and things like. There's a subtle, and there's different features of it that the professors, the more, I hate to say, modern professor who used discussion boards and stuff like that, which is something I'm not used to. I have to teach the way I learned to a certain extent, below those many years ago. Nevertheless, this is a supplemental outline. As you can see here, for a chapter about eukaryotic cells, we're getting to the cell next in earnest. And then lastly, I sent you effectively, I finalized the exam. And so we will conclude undoubtedly, likely very much by Friday, all of the exam material. It has a little bit on the cell, particularly about the way the membrane proteins are, and what we call the cell adhesion molecules that hold cells together. That's the end, even though there'll be more on the cell, but that sort of takes us into what's called movement across membranes, which will be on the second exam. But by incorporating, by going over that, it's amazing. We will be reviewing the other things, the stuff. So you'll almost simultaneously be reviewing material from the cell that's going to be on this first exam. That's kind of the way I arranged it. Down the road, I'll give you more particulars about it, but it's roughly 65 multiple choice, true, false, objective matching, that type of stuff questions. Because everybody asks me the same stupid question. Basically, you want to know, is it going to be an essay test? Objective means bring your number 2 or mechanical pencils. Nothing drives you crazier than, do you have another pencil? No, I don't. If you put pen, the machine doesn't read it, which upsets me no end. So file that under pet peeves. So all that busy work out of the way, we begin. So again, look for content, look for emails. I try to be as up on top of that as possible. And now we will conclude what we began earlier, and that is the end of this biochemistry unit, which begins with the nucleic acids. Again, they are the important point. Nucleic acids and amino acids. Nucleic acids are the polymer. Nucleotide, which is a small molecule, is the monomer. Very, very similar in the way that proteins, and fortunately, when it comes to DNA and RNA, we only have 4 to deal with. When it comes to there's only 4 monomers that constitute effectively the polymeric form we call the nucleic acid, either DNA or RNA, deoxyribonucleic or ribonucleic acid. It's different, again, with the amino acids, we had 20. Fortunately, you don't have to know the names of them, but you kind of get the idea. The other interesting thing, these also contain nitrogen. And why I make a big deal of it now is you're going to see down the road, particularly at the very end of AP2, nitrogen is a bit of a pain in the neck to get rid of. Our kidneys, probably the most difficult job, and the job that they have that is probably most vitally important, is getting rid of nitrogenous wastes. We have a very limited amount of nitrogen in bloodstream or in other waste products that we must get rid of. And the kidney does a very, very good job when it's working properly. Indeed, down the road, you'll see, how do we monitor or assess kidney function? We assess it by looking at levels of nitrogen compounds. One you'll learn down the road called creatine, and the other one is called BUN, or blood urea nitrogen. Here, a quick snapshot into whether or not somebody's kidneys are working properly. Okay, so I mean, that's all stuff that you'll probably see more so in AP2 than AP1. So I mentioned that as a side. Nucleic acids, again, nitrogen and phosphorus, because all of these have, and I wrote it up there, okay, when I was, and we do ATP at the end, so they have it in any nucleic or nucleotide, which again, it's a small molecule, has to have a base, has to have a pentose. The question is, pentose, if it's going to be this, a ribose, that's going to be something that's going to make probably evolve in some RNA, be a standalone in what we're talking about here, which you'll see shortly. So your choices with pentose are either ribose, see here, or we'll look for DNA deoxyribose. And there's a very subtle difference, but it accounts for the magic of DNA. So even though it's missing an oxygen, magically it allows DNA to form DNA. And that's just remarkable because DNA, as you will see, and you probably knew already, is a twisted ladder. It's called an alpha double helix. And so it's a spiral staircase and all of the rungs are equal. Okay, they're of equal width. That's the unique feature. That was the important feature. Ignore my bad handwriting over there for the time being. Okay. So DNA basically does two major things, all because of its structure. DNA, because it is held together, as you will see, by hydrogen bonds, can open and close. It's a biological zipper. It does it for two reasons. It opens and closes to make more DNA, exact copies, that it can use to make new cells. Effectively, it's a framework of what we call mitosis. The whole idea of mitosis, okay, is to make identical cells. It's we want exact copies. It's a little the term clone is probably incorrect, and we start looking at that a little bit more, we'll see why it's not exactly correct. But effectively, it opens and closes in such a way that you have and you'll see we have a strand in the old copy and a strand in the new copy, and a new strand kind of refreshes the DNA. Very, very useful. The other thing DNA does is it makes proteins. And it does so by expressing itself in the language of what's called RNA. There's a subtle difference in the way of the composition of these two molecules. DNA was, to give you a bit of the background, the historical background of this, everybody knew about DNA. Back at the end of World War I, when scientists began to study a lot of things, they knew that DNA was pivotal in how cells worked, how the body worked. They even knew that there were always equal amounts of adenine and thymine and cytosine and guanine. They were able to extract the DNA, and they found that they always had equal amounts. Not always the same. It wasn't like 90% this and 10% this. It was, but they always knew that the adenine and thymine mediums equal one another molecularly speaking. Other things that they knew. So they knew, and these are names that you do need to remember, that in terms of DNA, they had adenine and guanine. They're called purines. The illustration is better. Purines have a double ring. So again, adenine and guanine, names you have to remember. What are called pyrimidines have a single ring, and they're uracil, cytosine, and thymine. The difference is very simply stated that when we get to RNA, thymine is replaced by almost an identical molecule called uracil. They have one subtle side chain difference. That's it. And you don't have to know any of the chemistry of where the side chain is or why one is different than the other. Not at all. Just to know the basic building blocks, the ingredients. And we all call it the same thing. The A's, the T's, the G's, and the C's. You know all of that stuff. But what happened historically, they didn't know how it fit together. They couldn't understand the composition. And they probably would have solved the problem if it wasn't for another event that happened shortly after World War I, World War II. All the scientists started working for their individual governments and the countries that they represented in some scientific fashion. So during war, everything is dedicated to the war effort. That's the way it is historically. Particularly to World War II would be probably the best example of that. At the end of that, as soon as the scientists were free from doing the war effort, they went to other stuff. Okay, and what in England, an American and an Englishman with another researcher who were all credited with identifying DNA. Their principal names were Watson and Crick. I forget which one was the American, which one was the Englishman. And they were having a very difficult time. They had all this information, they couldn't put it together. And I'm sure I told you about it a little bit. Eventually, they magically put it together, came up with the structure, the famous twisted ladder, the right-handed double alpha helix. Voila, Nobel Prize, 1953. The magic of understanding opening and closing and all the things that really were the framework of what we call molecular biology. The problem was they didn't mention the lady named Rosalind Franklin, who really gave them the secret piece that they didn't understand. She was the one, she did what was called X-ray diffraction, which means she took molecules, okay, sufficient numbers of the same molecule, and X-rayed them. And from looking at the X-rays, you could determine a little bit of their molecular shape. The trouble was DNA was very unstable. It was always moving, and she couldn't get an accurate view. Is the ladder the same? You know, there's two of these, or there's the slats or the steps. She said, you know what I can do? I can freeze it. It's not like put it in the fridge, freezer. She put it like in liquid nitrogen, something extraordinarily cold, took the X-rays, discovered that the rungs, the steps of the ladder, were the exact width of a double ring and a single ring, a 2 and a 1, a 1 and a 2. She knowing in advance that there were equal adenine, thymine, cytosine, and guanine, 1s and 2s. She went, and the story is that she had done that in the morning, went down wherever they were all having lunch, and mentioned that I just discovered that this is the exact measurement. And incidentally, it's the same as if we combined a 2 and a 1. Watson and Crick and the other people went up and built the model, never gave her the credit. She was cited, she was given the honor posthumously, never won the Nobel Prize. They do not give Nobel Prizes to people after they have died unless it was awarded to them and they died before the actual presentation in Stockholm. A little trivia nugget for all your fans. That's how the story went. And it's a microcosm of what has, to a lesser extent today, it has gone away a lot, I'm thankful to say. But women in science and medicine did not get a lot of credit. And it's terrible, it is unfair, it is, I can't think of enough bad words to say about it that I would use during the classroom. But I could think of some other ones. So voila, that brings us to understanding what DNA does. It's all about, and this is what she helped discover, complementary base pairing. A's and the T's, the G's and the C's. They always got to line up. So, I mean, it's just when you have these two strands, you have the two rungs of the ladder, you have an A here and a T here, a G here and a C here. If you have A here and an A here, we have a name for that. It's mismatched. We call it a mutation. Don't misunderstand the word mutation. Mutation means errors in DNA. As your studies go on, you will see that we create mutations all the time. Every day, you're making mutated cells. We have mechanisms that prevent them from forming at all, from growing, or from creating mutations. We have so many safeguards that are there. Most mutations we make, you're going to learn about, are called silent mutations. They're a subtle error that doesn't impact anything, as opposed to a major mutation. Mutation doesn't mean you're going to have a third eye or an extra arm. No, that's a deformity. There's other things. So there's mutations. Mutations are just errors in DNA, period. Okay? If they call birth defects, we have another name for them. If they cause cancer, we call them carcinogens rather than mutagens, the name for things that cause mutations, okay? If they cause birth defects, they have a fancy name. They're called teratogens. None of which you have to know, but I want you to understand, people, it's important to understand the language for clarity, okay? Because we have a misconception. These days, they use words that are buzzwords and people click. So A's and T's and G's and C's, and this is what it looks like. The secret sauce, hydrogen bonds. By doing so, they know what they have. And this is where we call it the backbone. You have a phosphate, a sugar, and a base. On the other side, you have a phosphate and sugar in the base. Notice this is what you don't have to be so concerned about. We have a numbering system. There is directionality. And the easiest way to express it is these are identical chains. This is right side up. This is upside down. We have a fancy word for them. They're identical. If I had both black or both green markers, one is right side up, the other is upside down. Fancy name is called anti-parallel. And that's all it means. They're parallel strands. Parallel doesn't have to, it's not like geometry class. We don't have to have two parallel lines in a transversal. All those wonderful angles and stuff. I took that course too. Parallel just means they're going in the same direction. So, and with the beauty of that means when it opens and closes, whether you're making new, you're always going to have two identical strands, or whichever strand you use to turn around as a template, as sort of the framework to whatever proteins you're going to make, expressing as RNA. It doesn't matter which one you use. I mean, what they learn in the advanced classes like cell and molecular biology or genetics, they learn about the directionality, how it's assembled. They learn about, more of the intricacies of how that process goes on, where it starts and where it stops. And there's differences. One strand is made continuously, the other strand is made in fragments. Sometimes some of those fragments have to be disposed of. They have crazy names. Thankfully, you don't have to know, called Ozaki and Okazaki fragments. That's the kind of stuff, thankfully, being in this class, you're avoiding. So you should be very pleased about that. And so as I go back, what makes this the backbone is the relationship between the sugars and the phosphates. Even though you see it happen here, you did multiple of them. It's like this. I just use B, S, and B base. The other side, B, S, and P. I did this, heated it, and did this, and heated it. These use the little dots for high peak bonds. That opens and closes because it's a weak tone. Backbone that holds the molecule in place is this. The sugars and phosphates bond to each other and then to the next one, like this. The next one, like this. So it's the bonds in the sugars and phosphates that create what we call the backbone of DNA. And the only thing that differs is the bases. So you can see them here. And even though it isn't depicted particularly well in this illustration, the slats or those steps are the same. That's with DNA. So two jobs, open and closes, to either for replication, specifically we'll talk about mitosis more than meiosis, but even though it occurs in both, or, for this protein synthesis, making proteins. That's where RNA comes in. DNA is an enormous molecule. As you will learn, it is too big to leave the nucleus. It's not the only place in the cell where it is located. We'll get to that. But for now, it is too large to leave it. The nucleus has little pores that you will learn about. Okay, that's how building materials come in, waste products go out, and and the opening is fairly large. It's large enough for RNA to get out, but not DNA. So in order for DNA to express itself in terms of making proteins, it has to rely on another molecule, another giant molecule, another nucleic acid. This one is RNA. So the only thing that, again, is different is the sugar, ribose, and the fact that two almost look like identical twins molecules, uracil replaces thymine. So we don't have the A's, the T's, the G's, and the C's. We have A's and U's and G's and C's. That's the big difference. And down the road, if you get a chance, and it's more in micro for many of you, that you look at this. The genetic code is in terms of RNA. So you see U rather than thymine. So if you studied that before or seen, the typical genetic code, the 64 possibilities. It's in terms of uracil. It's got fancy names called codons and anticodons and triplets and stuff like that, none of which we have to get to in this. And depending on if you've taken micro, you had the micro already. Did you? Yeah. Depending on who teaches it, they may have gone into that. When I do micro for health science and nursing students, you got better stuff to talk about. I'm annoying anyway. If you're stuck having me again, same bad jokes, you're obligated to laugh. There could be someone who hasn't heard them. You're under duress. They nearly blew that game last night. I'm throwing stuff at my iPad. It's like the Pirates. The Yankees had a 10-1 lead after like 4 innings, won 10-9. Thankfully, David Bednar came in and only gave up a home run before he got the save. Try it. Nevertheless, so what does RNA do? Okay, it has to carry the message. What it does, the first part, it has 3 different types. There are more than that. Okay, thymine replaces uracil. There's actually what they call messenger RNA is not just plain messenger RNA. Again, it's not something we typically do in this course. Something that we talk about again in micro. It has to do with what our cells have. They actually take a very long strand of messenger RNA and kind of chop it down so it just gives you the genetic information. But that's another lecture entirely. For our purposes here, what messenger RNA is created from DNA in a process called, and this is an important name, transcription. When you transcribe something, it's an exact copy of the message. That's what's difficult. It's not like if you're listening to me this lecture, you're taking notes maybe. You're not writing down every word I say. Now, if this, and I know that these voice programs that we use today are not 100% accurate, but effectively it's trying to transcribe. It's trying to record exactly what it is. And the anecdotes are a couple of them in the hospital. We used to do an operative report right after an operation. We would I haven't operated in 25 years. We would pick up a phone or have a dictating machine and you dictate the report. I did this, this, and this, an exact report. Someone would get that recording. They do a transcription and you sign off and I went into the hospital chart. I know everything is in digital age, different. So effectively, transcription means it's making an exact copy in its own language, its own form of molecular shorthand. In the old days, you'd write, my mom did this. My mom was a professional secretary. She'd write in shorthand. She'd take the letter, she'd type it up, bring it in for the boss to sign, they'd mail it. That's the way it used to work. So messenger RNA, whole idea is to carry the transcribed message, hence we call it transcription. It takes it to a ribosome. Ribosome is made, as you will come to find out, of ribosomal RNA and protein. It's a platform specifically constructed to assemble amino acids, chain them together to make polypeptides and proteins. So that is the platform. That's what ribosomal RNA is about. And at that platform, the combination of the messenger RNA and the ribosomal RNA will assemble those polypeptides and proteins with the help of yet another RNA called transfer RNA that brings the amino acids that are out in and around the ribosome to that ribosome so they can be chained together. You'll see how this works down the road. I'm not going to go into the gory details of it. It's a very elaborate process with different sites and all the things. You don't have to go. I just want you to get the general idea of what those things do for now and for later. That process, all of that process, taking the transcribed message, taking it to the ribosome, bringing in the amino acids, is called translation. You're taking the language of DNA and the transcribed language of RNA and basically assembling protein. That's transcription. That's what it does. I didn't get a lot out of this. It's long. It goes in. I'll kind of show you. When you begin to look at this, DNA. Yeah. So it shows you the different molecules and blah, blah, blah. Notice on the bottom, as you look at it on the right, uracil and thymine. They have one subtle difference sort of at maybe 10 o'clock, if you look at that, at the two molecules. Those actually have numbers, and that's what they go into. Why do so they're almost identical, it's a subtle difference. And what we do, and why I go into this, I hear you. Why this becomes important, why I'm spending extra time on this. We have taken in science, we take these molecules all the time and put substitutions on them for different purposes. One of the earliest treatment for breast cancer was uracil. They took uracil, and in this position, which is called the 5 position, just go with it. They took if you look over there in the periodic table, all the halogens, fluorine, chlorine, bromine, iodine, when you put them on a molecule, they're somewhat toxic. And this one was they put a fluoride group, and that was called 5-fluorouracil. That was one of the early treatments for breast cancer. It's called an anti-metabolite. So what happens when the cancer is assembling all these DNAs, and particularly the RNAs, it picks this up and it effectively poisons that cancer cell, and the cancer cell dies. The whole idea, and we use them in other forms. We take adenine, make substitutions on that, and if you know somebody had a cold sore and they got a prescription for Valtrex, pretty common and a very good antiviral, works the same way. So cancer chemotherapy and a lot of antiviral therapy we call nucleoside, basically nucleus sort of look-alike drugs, the way I call them. So that's why, and that's, going forward, I want to give you some context as to why it'd be good to have some knowledge about what those things are. Because many of you, when you go into clinical fields, as most of you are going into, you're going to see this and go. When you get into the pathophysiology and the pharmacology of it, this gives you a certain degree of assistance in being able to understand it and remember it, I think. And then the rest of this goes into these numbers. So they're showing you how they bond together and blah, blah, blah. See, they are all 1, 2, 3, 4, 5. There's a way, when I had the upside down and the right side up, the way that they're assembled, there's what we call 5' to 3' and 3' to 5'. Thank goodness I don't have to teach you about it. I already understand it. But it allows the molecule to be assembled. Is all that they're trying to give you out of that. So it's not really that important. Unlike some molecules, the small molecule is vital as well. And there's one of these, again, nucleotide. Nucleic acids, DNA and RNA are polymer. Monomers are called nucleotides by definition. They consist of, again, a base, 5-carbon sugar, and for these, as far as I know, they're pretty much all ribosomes that really have molecular activity and phosphates. This is one you're going to see 2 of them during the course of this term. You're going to see them adenine and ribose and guanine and ribose. They're both going to be bonded to phosphates. The combination of adenine and ribose is called adenosine. Combination of guanine and ribose is called guanosine. Down the road, toward the end of the term, we're going to talk about what are called neurotransmitters, how the nerves communicate with one another in parts of your brain. Every morning, I make an attempt to not let adenosine's message get through. Adenosine is a neurotransmitter that makes you tired, rest, relaxed, plays an important role in pregnancy, all very important. I block it every morning with caffeine. How does caffeine work metabolically? It blocks adenosine receptors. It makes us awake, or it makes us more awake and alert. And excessively, so much so it makes you jittery at times, or excessively, so much so people are allowed to sometimes have close heart palpitations. Nothing unusual about it. Colds work the same way. Tea works the same way. All those Red Bull and all those related energy stuff work the same way. So you got to be careful with those, just from a standpoint of consumption. But the single most important molecule, other than in this category, other than DNA and RNA, even though is ATP. Adenosine with 3-phosphate. Phosphate is a PO4 with a minus. You can see it as a negative charge. And you have 3 of them together, and we always draw this sort of squiggly bond to indicate it's a high energy bond. Because when we separate the phosphates, we can release that energy and use it to do work in our cells, which is very advantageous. So in this case, you're going to see, I commented on it a little bit before. We open and close, we break apart and reform those phosphate bonds between ATP, adenosine triphosphate, and ADP all the time. So much so, if we added them up, all of those molecules, ATP and ADP, and added have untold trillions and trillions upon trillions that we utilize every day, they would weigh as much as you do. Imagine that. That's how much energy we're constantly breaking down and storing. This molecule is magical. It's magical in its shape. And what's neat about it is the fact that those phosphate groups, I don't know, if you can come up with a better analogy, I'll be happy to use it. I think kitchen magnets. Don't look at me funny like that. Someone in this room, not just me, has taken kitchen magnets and had played with them to the point where they would either see, you're guilty, would either stick together or pop apart. It was fun. All that. Negative charge. You're used when you have the same charge, it takes extra energy to hold it together. Imagine when you took a third one. You let go and boom, it almost pops out. Love that. Put them on the fridge, push them around the kitchen. Mom's yelling at that. It's my favorite magnet. Don't do that. It's good stuff. Okay, phosphate groups are because of the negative charge. So that, and we use every bit of them. The adenosine, the adenine and the ribose, we use, as I said, as a neurotransmitter. When we add a phosphate, creating adenosine mono for one phosphate, you're going to see inside the cell, it directs certain activities when we get to how it is used inside the cell. ADP and ATP are constantly exchanging places. When we need energy, we release the phosphate. When we want to store energy, we bond back. And so we release the energy, we release the phosphate to do work. When we have extra, when we've used it all up, we put them back together. The trouble is they only really stay in one place for about a minute. Then you have to store them if you haven't used them, and they store them as fat. That's the big problem. So when we talk about energy utilization, it's like calorie consumption, which are really energy units. So if they say you're supposed to eat 2,000 calories, no more. If you eat excess calories, you're going to end up with storing more fat. If you want to lose weight, you do less calories. That's the simple basis of dieting, and then the nuances with different things, different ways to approach that. In the old days, calorie counting and Weight Watchers and stuff. All that's been around for a long time. Nevertheless, that's kind of the basis of all of this. So that ATP can be transferred to other compounds so they can use the energy. And it converts ATP to ADP, and we can even lose the second one. For instance, something we're going to look at when we start the next chapter are specialized openings that open and close. We call them transport channels. And I demonstrated a little bit with those stupid papers that I used before. Opening and closing the transport channels. One of those is for glucose. There's always glucose outside the cell. It always goes inside the cell. What happens as soon as a glucose molecule goes in, remember I told you about activation energy. We immediately take an ATP and tag that to a glucose. We create, it's a fancy name, it's called G6P. Glucose, we had a phosphate in what's called the 6th position. That makes it more water soluble. And the fact that we added the phosphate means it's no longer glucose. In living systems, once you change a molecule, it's not the same. So every time glucose enters the cell, it immediately gets changed. That's why there's always more glucose outside the cell than inside the cell. So all you have to do is open the door and in goes the glucose. That's what insulin does. So it works. You don't have to know this stuff. I'm trying to give you context. Okay. And that, when our students do the advanced stuff, they learn about that particular pathway. It's got a wonderful name. It's called glycolysis. So that's why it's so important to utilize it. When we get to seeing how muscles work and how the muscle actually works, what the ATP does, you're going to see when we lop off a phosphate, it does something to one of the motor proteins to make it ready to fire. See, all of these things are part of it. So we were very conservative, maybe, I don't know if it expresses it as well. We use everything we can. All the ingredients we have, we try to find a use for. And this is kind of the understanding of how things work in the body. Constantly energizing things. Energy is basically, I said, that's part of that energy of activation that enzymes work with. So you begin to understand how this all kind of comes together, even though you probably won't be using it a heck of a lot. We have this is a different kind of a channel. This is a membrane protein that won't open without energy, because there's more stuff outside than inside. And so in order to kick stuff outside, we basically have to add energy to open that. It's got a fancy name. They're called pumps. We'd be sick and tired of hearing about them. Okay. How do we get a muscle to contract? We use ATP. It's interesting. They have sort of a spiral form, the way they're constructed, smooth muscles. So they kind of bulge a little bit. Okay. It has to do with the shape of the muscle and the fibers that are in it. And then even putting molecules together, just giving you some examples that are there. And thankfully, that ends that biochemistry chapter. I'm sure you're delighted. God, the first time anybody laughed. And even one day, somebody's going to send me an email other than saying I'm sick. And I'll be happy with that. Someone actually asked me a question. Be inquisitive. Be courageous. Sorry, don't get me started. I'm showing you, this is the one I sent you. When we get to this last slide, which is wherever this is sequentially, slide number 39 of that's the last slide, and that's the end of it. That's what concludes. The first is just going to be on how these cells join together and the role of proteins in the membrane. I just want to review very briefly what I sent you when I was away. A lovely quiet river scene while I dictated this to you. My wife was thankfully sound asleep, and I was not disturbing her. Next spring, we're going to stay in a yurt. You know what a yurt is? See, we're going to stay, but we're glamping. We're not staying in a yurt. I figured this is outdoor plumbing. This is a glamp yurt. You go, girl. See, when I got married, I told her my idea was roughing it, was going to a hotel without room service and in a room that didn't have a hot tub already in it. That was my idea of roughing it. That was my first wife's idea of roughing it. Explains why I have no money. Moving on. So I gave you this. This is the one I sent you. Listen, I'm not going to go over cell theory or cell diversity. There's a whole lot of different cells, and their shape plays a pivotal role. Okay, whether it's red blood cells carrying oxygen, whether here are skeletal muscle cells, you're going to see all the way that they're arranged, fat cells carrying stuff, and the really hard ones to deal with, nerve cells. Because there's so much intricacy to it. So the shapes all tell us something. So that's the idea of basically the introductory nugget for the cell. Yes, they all have a membrane, and that's really the first part I'm going to reinforce. Okay, it's flexible, but it's permeable. Living systems, by definition, every single membrane is a semi-permeable membrane. It's a boundary, and some stuff can get in, some stuff cannot. We call it selective permeability. It's the hallmark of living membranes. With very, very rare exception, there's even those are permeable, but there's some that are really significantly, limitedly so. And we will get to those, particularly in the nervous system. Cytoplasm, okay, the stuff that's inside the cell, pretty consistent. It's either liquid or gel-like. Some of it's more solid. Doesn't really matter for now. And yeah, sure, the nucleus, the control center. We'll deal with all of these in a little bit more detail. This is a useful generic illustration that points out, to some extent, the complexity of the cell. This is years ago. First time I had a teacher, whatever grade this was in elementary school, we're made of cells, here's a cell. That was it. That's about all they knew. They had no idea of all the stuff that goes on. But really, what we do is you did a little bit of looking at things microscopically in the lab. As people get more experience, when their job is looking at that, if they're a doctor called a pathologist, where they have to look at normal and abnormal cells and make diagnoses based on the cellular or the tissue structure, you look for differences in the individual components. This is the nucleus is quite big, it's not that large, but you're trying to do our little pores that the RNA can get through the DNA is too big. This is called endoplasmic reticulum. All of these membrane channels, all of these organelles are connected. It's called the endo or inside membrane system. And basically, anything you make that the cell is going to use is so subject to the interior of the cell. It's a harsh place. It's got chemicals, it's got acids, it's got bases, it's got salts that would ruin the protein or ruin the material. So anything like that is held and encased in membrane containers, whether it's going to be it out, whether it's going to go into the membrane, whether it's going to go to the organelle, wherever it's going. So it's a very, very harsh environment. It's the beauty of this system. So it's like continuously being protected by membrane. Membrane protects it and protects it from other things that could impact it. And it basically creates an area or a platform to do whatever you have to do. Proteins are really big. They have to put different heads and tails on them, packaging. It's like you get a package from Amazon. Okay, I mean, these days they do it. They'll just throw it in the original box and say this is okay for shipping and put a label on it. But if you bought a new laptop, it's got to come with all the plastic around it and the bubble wrap and the packaging and all that stuff. Your body does the same thing. Okay. Basically, it's a useful analogy to the way those things are done. So we'll look at the roles of all of these things and what they're able to do. But this speaks to the complexity and different arrangements of different cells give those who study them a clue as to how they work. More on that down the road. Intra and extracellular. Intra for inside, extra for outside. I mean, I commented on that. These fluids are similar but not the same. Sometimes it's different amounts of proteins. Sometimes it's different components that are there. For instance, in plasma, okay, you'll have one level. Interstitial fluid between the cells, will have other types of chemicals in it. Cerebrospinal fluid there are certain things that we find in there that shouldn't be in there. If we find a white blood cell, which might inadvertently be in the plasma, it's a bad thing, because that might point to somebody who has meningitis. They're not supposed to be there. And indeed, inside the cell, it's going to have similar makeup, but different, particularly with regard to the electrolytes. And that's really the focus point, I think, about understanding how cells conduct impulses, how nerve and muscle cells work. It's called resting membrane potential. We're going to get to that. Again, saliva and mucus are both made by the cells, except one has a little more protein. Mucus, it's sticky. What it does is it's either protective or stickiness that plays things. And you talked about this matrix in connective tissue. And so the membrane itself is this, and that's the beauty of it, okay? The controlling the flow in and out. So thankfully, you don't have to go crazy about how it's assembled. If we take phospholipids, and why I spend time talking about phospholipids and throw them in water, they naturally form a bilayer, a membrane. It just so happens in our bodies that occurs in proteins. And that's why I went into all the stuff about folding the proteins, things like all the different layers and fat soluble, all the fat soluble components, amino acids kind of come together so they can insert themselves. They're compatible with the interior of the membrane, but they can change on the outside and change on the inside. Okay. And that's where you come to that fluid mosaic model. The fluidity comes from cholesterol. That allows it to open and close, change shape, envelop things, push things out, that rapidly retain the overall integrity of the cell. So, and we'll look at everything, the sugars that stick out on the outside that are a signature or for places to attach, or all of those types of things, and the cell junctions, and that'll be the last part of the exam. So I will go into all of these. I commented on them before, but I'll start here with all the classes, go into these. And like I said, by the time all is said and done, it won't take very long till we get through all the mechanisms of these and the joining factors, and hopefully we can get it all done on Friday. Thank you. Bing.